Hello, my name is Ross Cagan. I believe I'm online now. This is my first time doing an online talk, so it's as if I'm giving a talk to myself, which is fine. My name is Ross Cagan. I am a professor and associate dean, and also I am director of the Center for Personalized Cancer Therapeutics at the Icahn School of Medicine at Mount Sinai. And actually, as declarations, and as I'm hoping my slide will show, perhaps, that you're right. So I'm also, this is my declaration. I am also co-founder and on the SAB and the board of directors of Medros. Nothing I'm going to talk about is directly related to that company, but I do want to disclose it. I'm also editor-in-chief of Disease Models and Mechanisms, and I just want to mention that because I hope everybody here is considering sending their next paper there. Okay, and this is the outline to my talk. So I am a Drosophila biologist. What I'm going to talk about today is how we use flies to develop new types of drugs and new approaches to cancer therapeutics. My laboratory also works in diabetes, and we both explore and develop therapeutics for diabetes, but I'm not going to talk about that today. Pick one topic and stay with it. So I'm going to talk about really two approaches that we take. One is I'm going to talk about how we use flies and chemistry to develop complex drugs, and I'll explain what that means, mainly polypharmacology. And then I'm going to switch, and I'm going to talk about how we use Drosophila in ways that you really can't use other animals to develop complex cancer models. And I'll give an example of why that matters, why complexity matters. And finally, I'll finish my talk by describing a center which we just opened the doors on. I am the director of it. It is the Center for Personalized Cancer Therapeutics, where we use basic research tools, including Drosophila, as I'll explain, to more precisely model the complexity of the patient's tumors, and then using high-throughput drug screening, develop whole animal-based therapeutics for those patients. And a lot of my talk will be explaining why that matters. Let's see. Let's push this. Okay, so I have many slides that I often start with when I give talks. For this one, I thought I would put out this. I see it's actually a merger of two slides. That's okay. And what this is showing is the cost for the cost by pharma from 1950 to today to develop a successful drug, and that's cost per drug. Drugs per $1 billion. It went from 100 drugs per $1 billion to well less than 10 drugs per $1 billion in 2010. Okay, and this is inflation adjusted. Now this looks very impressive, the way this curve is going down, but actually it's more impressive because if you look carefully on the left, you'll see it's actually a logarithmic scale. In fact, we are getting worse in a logarithmic way. Now what's the problem? So, and the reason I bring this up is because I'm really answering in part the question, why Drosophila? And part of my answer is that we have many great models and a lot of very smart people working on it, but it's obviously a difficult problem, cancer and disease in general. And in fact, we've not been very good at developing successful drugs. The hit rate is quite low. And sometimes I'll show the success rate of clinical trials. And for cancer, it is commonly in the single digits for success rate. That's ridiculous. So what's the issue? And as I see here, these slides are merged. So I'm relatively new to the cancer field. And so coming into it, I've sort of been thinking about what are some of the things that have been issues and what are some of the assumptions that we've made in developing therapeutics. One is there has always been a push, by myself included, to try to simplify the disease. That is, can we find commonalities between patients? Initially, it was trying to find that one driver that's important. Maybe it was the driver you studied when you were a postdoc. Maybe it's a driver that you were taken to by a screen, and then you focus on that one gene. and you sort of beat that to death, maybe even the whole pathway. And the idea is then if you drug that, that should knock out the disease. The problem with that, of course, is that it's a complex disease, and I'll make that a finer point when I show you some of the data that we have. And I will show you examples where actually when you account for the complexity, drugs that work in simple disease models don't work in complex disease models. And I'm actually going to really push that issue of embracing complexity, even potentially to the point where we embrace as much complexity as we can. So in other words, we're trying to push the opposite direction. How much complexity can we pour into our models? Also, there's been an assumption, not unreasonable and not always untrue, actually, sometimes it's true, that the driver of the disease is also the best therapeutic target. And there are examples of this. EGF receptor is an important driver for certain cancers, and it has been a terrific therapeutic target. But in fact, in most cases, when you, I think, in most cases, genes that are so fundamental to the biology of the cell that when you just change them a bit, they can cause dramatic problems like cancer, diabetes, and so on. These are so fundamental to the biology of the cell that when you drug the patient, when you drug those down in a patient, that can cause toxicity. And in fact, it's been shown in a number of studies, recent studies, that targeted therapeutics are overall more toxic than the more basic chemotherapy. And so again, a lot of the drugs that are successful in the marketplace, if you ask what do, they don't hit the central pathways that we all know and love. What they really do is they sort of nibble around the edges of the network and sort of push that network back into some semblance of normalcy. And then we can get along with that push, but in some cases the disease cannot. Also riffing off this a little bit, there's the issue of single versus multi-targeted drugs. This has been a debate in the field that has heated up. Actually, when I got started, there was no heat. It was pretty much everybody was for single targeting drugs. I'm a big fan of multi-targeting drugs for reasons I'll explain. And finally, cell-based screening versus whole animal screening, I would say that they are simply different. Some of the differences and advantages of whole animal screening are obvious. Some of the disadvantages are obvious. You can't screen as many. It's less precise. But in all the whole animal drug screening that my lab has done, we've really gotten to think about some of the advantages of it that we hadn't really considered when we started this. And some of the key advantages are that when we look at where are these drugs actually acting, most cases the drug hits multiple targets. Some of those targets are actually on the tumor, on the sugar uptake aspects of the cell. But many of the activities of the more successful approved drugs are actually elsewhere in the body. So they act on the liver, they act on the muscle, which then somehow acts back on the tumor. And in fact, I suspect that in many cases, when you have two drugs that are supposed to hit the same target, one is successful in the clinics, one is not. It may simply be because those so-called off-target effects are in fact important, but important elsewhere in the body. Because of course, when you take a drug, you're drugging the whole body. And that means that you'll identify a class of drugs that you will not identify in a cell or in vitro-based screening. And I would argue that that is a potentially successful class, but the onus is on us to show that. So let me move forward. So a few words about Drosophila. Here we go. A few words about Drosophila. For those of you who haven't thought about flies in a while, maybe since high school or college, and you probably remember there are white eyes and red eyes. And if you cross them together, somehow a couple generations later, if you do the Punnett square, you can figure out how many white eyes you'd expect, all that stuff. So flies have come a long way from this. To summarize what you can do in a fly, which has flat out the most powerful tools of any multicellular organism, model organism we currently have, you can turn any gene or genes on or off in any cell, not cell type, not just time, but any cell at any stage you wish, period. Okay? And almost always it's with off-the-shelf reagents that you just order in from various stock centers. And that, for a cancer, is very powerful. So we can begin to think about making a 3, 4, 5 hit model or even beyond. And as I'll explain, we've really been pushing the technology. It really changes what you can do. So other things about flies, most of the basic structures that we have are conserved in flies. They have conserved epithelium, so my lab really focuses on solid tumors. In fact, they're really the only simple model organism that you can do high throughput screening in because they have conserved epithelia. Simpler organisms really don't. They have most of the organs we have. They have hearts, they have livers, they have kidneys, and so on, and my lab studies them, as do many others. The majority, not all, but the majority of genes that drive disease in us are conserved in flies. And in fact, much of what we know about the major signaling pathways, I shouldn't say much, but a lot of what we know about the major signaling pathways actually comes from flies. And that's why these pathways have crazy names like notch, hedgehog, wint, and so on. These are actually descriptions of phenotypes you see when you knock them out in flies. And so, as I mentioned, I would consider flies to really sit in the cancer sweet spot. Animals that are more complex, like zebrafish and mice, they're wonderful, but they're not very good for high throughput screening. Simpler organisms like C. elegans, for example, don't really have epithelia the way we do, or not exactly the way we do. And so you don't see a lot of solid tumor work in them. Flies really sort of hit that sweet spot. They have a 9-day life cycle. They're very cheap. You can grow lots of them in a single vial. So they're very easy to work with. There are plenty of disadvantages in flies. I would say the major disadvantage is in the details. So we try to think of flies as putting us into new areas, new chemical areas, new therapeutic areas, new ways of thinking about disease, but we don't obsess with perfecting a drug in flies or obsess over working at the tiniest details of the mechanism in flies. Then we actually, once we have the question clarified and we're put in the right place, then we move to mammals and ask, is this true in a mammal? In most, but not all cases, it is. Okay? And they do have other differences. They do have an immune system. They don't really have an adaptive immune system in the same way we do. They don't have immunoglobulins, for example. And that's important to think about some aspects of inflammation. So there are disadvantages as well. And so what we try to do is that we try to leverage those things that are strengths in flies by choosing carefully what diseases that we study. Okay, so I'm going to start with a model that we developed some years ago. This is actually my entry point into disease, my lab's entry point. We were actually a development lab, like most Drosophila labs, neural development. And after a conversation with a colleague of mine, Paul Goodfellow, a human geneticist, who was studying a disease called multiple endocrine neoplasia type 2. And the major problem in these MEN2 patients is a thyroid cancer called medullary thyroid carcinoma. Some of you may be familiar with it, some not. It's a relatively rare, it accounts for a few % of thyroid cancers. However, it is one of the more serious ones, along with anaplastic thyroid carcinoma. It is one of the very few types that will actually metastasize and can eventually be fatal. The other interesting thing about MTCs is that the majority of them are driven by an activating mutation in the RET receptor tyrosine kinase. And I have here on the screen a high-resolution crystallographic structure of RET. It's a transmembrane receptor. It binds ligands like GDNF. It has a cofactor that it complexes with. Upon binding, it dimerizes and activates downstream signaling pathways like RAS, SRC, Jun kinase, and so on. Many of the pathways that we all know and love in the signal transduction field. It's a classic RTK, but these patients have activating mutations in them. Some are born with it, a quarter of them. Actually, the majority, it arises spontaneously. And these mutations spontaneously activate the pathway. So it's on all the time. And although RET is broadly expressed in your body, it really, its major defect, as I said, is in the thyroid, in the parafollicular or C cells in the base of the thyroid. And the result is overgrowth, proliferation, eventual metastasis, and so on. So how do you model this in a fly? Well, flies don't have a thyroid. I just told you they have many organs, but they don't have thyroid. Although, to be fair, they have a very skinny neck, so we may have missed it, but I don't think so because they don't make calcitonin. the other things that C cells make. So we target it to the eye, in part because that's what my lab studied as part of the nervous system, and in part because flies can live without their eyes. So I'm going to talk a fair bit about these rough eyes and so on. And so let me get you used to it. So here in the middle panel, I don't know if my cursor is showing up everywhere, but here in the middle panel, we have a wild type eye, and you can see that the eye is nice and smooth. Okay? On the right side, we put oncogenic ret, either human or in this case fly ret. They both work equally well in a fly, by putting a single point mutation in it, in the intracellular domain. And I hope you can see that the result is a really bad looking eye. And if you look closely, you'll see there's little bulges coming out of this eye. These are actually tumor-like growths in it. What you can't see, if we cut that open and look underneath, you'll see cells leaving there, classic metastasis-like behavior. You see switches in cell fate, but you also see compensatory apoptosis. A lot of the things that you see in a real tumor, senescence, etc., we see in this tumor. And in fact, my lab has really taken this apart, and we've laid out, I think, most, if not all, the major pathways that are triggered by oncogenic ret to drive this tumor. We use classical genetics to really walk through here. We found 140 genes that drive this disease, okay? Or at least can drive this disease. Now, so we know quite a bit about it, but what we did was probably more interesting is shown in this slide. Now, I did try to load a movie here, but unfortunately it didn't load. But if the movie on the right was working, what you'd see was a very slick-looking presentation where essentially what we do is we grow flies in a 96-well plate format. So it's a little demeaning to the flies, but it works. We have robotics that move food and drug into a deep 96-well plate. The robotics mix it, and my lab has 4 different chemical libraries that emphasize druggable space. We then we have a modified FACS sorter from Union Biometrica that spits 10 embryos into each well. Okay, so you have 960 embryos in this 96-well plate. We put an oxygen permeable lid on top. The flies hatch out, they eat the food, they eat the drug, and then we do basically a phase 3 clinical trial right there in the dish. Okay? And we have actually, we generally set up the model to either have a very rough eye or even set it up so the tumors are so aggressive they kill the animal. And then we just screen. In most cases, we look for rescue from lethality. And we just screen for drugs that unkill the fly. And it's a very easy score. You just look at the plate. If there's anything moving in there, that's your drug. Okay, it's wonderful. It works well. And my lab is very good at this. So we've really pioneered this as a whole animal drug screen. So will this work? Well, in the early days, we screened smaller libraries, including libraries that contained what was in retrospect approved drugs, at least it is now approved. So one of the hits, in fact the major hit that we got from our initial screens was, again, on the left you see a normal eye. In the middle you see an eye on oncogenic ret, expressed just in the eye, and you see the tumors coming out of it. And on the right you see oncogenic ret targeted to the eye, but now the fly has been eating orally in the food in its well, has been eating a drug called ZD6474. Okay? And I hope you can see that in the presence of the drug, it completely blocks the effects of the oncogene. And that eye, it's not quite normal, but it's basically a normal eye. And we really didn't have another drug that could make that eye look so good without basically killing the animal. This drug could bring the eye all the way back to normal at a 30-fold lower dose than was required to kill the fly. So in other words, if you were a fly and if what was in your food is what's in you, then I would say the therapeutic index is 30, which is a pretty amazing therapeutic index for an anti-cancer drug. So I have to say I got very excited about this. This was when I was at WashU Medical School. I went running around the medical school saying you can cure these patients, you should just give them this drug. So apparently you can't actually take drugs from flies and give them to people, which was the first of many disappointments in this. But to cut to the chase, at this time there was also some really important work that was done by Massimo Santoro, who's a biochemist and cancer biologist over in Naples. And he showed that this same drug, ZD6474, was an effective RET inhibitor. It was not originally designed as a RET inhibitor. This was made by AstraZeneca to attack a completely different receptor called VEGF receptor, but that it could successfully repress RET and other targets as well. And they were able to show in a xenograft model that it was effective. So based on all of this data, Sam Wells took this drug into the clinics with AstraZeneca. So AstraZeneca was on our first paper showing basically these images. Took this drug into the clinics, and to cut to the chase, this was approved. So this is a CT scan of an early patient, but actually I'm not going to get into this. I have more to talk about. So it was approved as the first chemotherapy for medullary thyroid carcinoma. Okay. So honestly, that worked better than I expected. So I switched my lab out of development, full on into disease, and moved to New York to the Icahn School of Medicine at Mount Sinai. And we've really focused completely on disease, mainly cancer and diabetes. So what have we learned from this drug? What is it doing? Is it just inhibiting RET? Well, our genetics actually told us, and I think I'll show you a little bit of data on that having a clean RET inhibitor is actually not the way to go to successfully knock this tumor out, because flies, probably like us, we need RET in other parts of our body for normal homeostasis. So what you want to do is dial RET down, but you don't want to dial it out. And that means you're going to need to hit other targets as well. And in fact, we now know, based on in vitro kinase data, that ZD6474, which many of you know as vandetanib, is basically the world's worst kinase inhibitor. It hits a lot of things, a lot of kinases, and it hits them poorly. But what's magic about it is it hits them in just the right ratio, that the fly can take that hit. As it turns out, we can take that hit. But the tumor has difficulty selecting around that to get around that. And in fact, patients who are on this, those that do respond, and about 60% plus respond to this, they commonly respond for several years before showing resistance. And we think that's in part because it hits so many different targets that the tumor has difficulty finding a road around all of those different hits. And so that really taught us a lot about what we feel we need in a drug. Lower specificity, hit multiple targets, but do it in a whole animal system, because it would be almost impossible to predict out which things you would need to hit without at least getting a constant whole animal view of what's toxic. However, just building low specificity kinase inhibitors out of flies is very difficult, because the chemists will complain fairly that if they don't know what's the important activities, they don't know what parts of the molecule they can manipulate to optimize it for people, and so they can't work with it. Okay, so the question is, could we come up with a method to do what we call rational polypharmacology? That is, build a drug that hits multiple targets, but in a rational and whole animal way where we monitor everything that's important. And that takes us here. And this was a collaboration with a colleague of mine, Cagan, a really fantastic medicinal chemist over at UCSF. Really one of the very best medicinal chemists, especially specializing in kinase inhibitors. And this was work done with his postdoc, Arvind Dar, who we now stole and is now a professor at Mount Sinai. And really my postdoc, Derta Das, who's spectacular, who is now just deciding where he's going to move into his first PI job. And this was, I think, these guys did really an impressive job. And what they did is as follows. So no more rough eyes, no more tumors in the eyes. They now set up a system where they expressed oncogenic RET in several parts of the fly in a way that basically killed the fly. Okay, so the fly is now riddled with tumors and dies. And as I mentioned, when we do drug screening, we actually like death, because then we can rescue from death, and that's all good thing. The flies are a little up and down about death, but it works for us. So they set up the system where they express RET in several parts of the fly, and they calibrated it, and you can see this here in this bar, so that half of the flies survive to pupation, okay, and none of them survive to adult, right? So it's embryo, larva, pupa, adult. They make it all the way to pupa and die because the tumors kick in around that time, and none make it to adult. Now, we didn't have a clean RET inhibitor, so Cagan's lab actually generated one, which is the cleanest I'm aware of, At the highest dose we can give that RET inhibitor, called DL06, you can see that it doesn't do much to help the animals before, and at any higher dose, it essentially kills all the animals. Okay? So coming up with a clean RET inhibitor did not seem promising. So we screened a library, and this library was a special library. The reason I actually went to Cagan is his specialty is building libraries of drugs that target multiple targets. In their case, for research purposes, but the good news is they built it from a library that has druggable properties. So we screened this library, the AD series, so-called from Arvind Dar, who built the library. And we got one hit, which I'll call AD1. And you can see why we like AD1. And that's here in the middle. I think I don't have a pointer, but whatever. AD1, you can see that you have a higher level of animals surviving pupation, and finally, we're actually getting adults out. Okay? So we're very excited. We're getting like 30% adults. Very good. Very good. Now, let's see. I think this will work. Yes. Something more should happen. There we go. Okay. So you can tell that we've patented this drug because I'm showing you the structure. So here's the structure of AD1 on the left. Now what's interesting is we looked at close analogs of AD1, other drugs that were almost identical but with small changes. For example, AD2 is identical to AD1, but it's missing that trifluoromethyl group that you see in red on the upper left. And that small change causes AD2 to become highly toxic. We're actually getting fewer animals than no drug at all. AD3 is identical to AD1, but instead of a single methylene group shown there in red in the middle between those two rings, it actually has two bonds there, and that's sufficient to kill a lot of the activity. It's a little bit of activity, but nothing impressive. So, what can we learn about these drugs from both the drug that worked and the drugs that didn't work, and can we build a better AD1? Now, what I didn't tell you, mercifully, is that we had done this large genetic screen. Actually, I did mention this. We got 140 genetic modifiers, things that drive RET-based tumorigenesis. And the majority of them fell into three basic pathways, which I'm going to summarize here. RAS signaling, which is dominant for proliferation. SRC signaling, which my lab spends a lot of time looking at, which drives metastasis. Okay? And PI3 kinase signaling, which does a number of things and sort of pumps the whole system up. Okay, so it contributes to both of these pathways and others, and also regulates metabolism. Now, the rate-limiting enzymes in these are RAF, mTOR, and SRC. Okay, and those happen to also be the three druggable enzymes in that pathway. All right, these are kinases: RAF, mTOR, and SRC. So let's look at it in human in vitro kinase panel and ask, what does AD1, 2, and 3 hit regarding these, actually 4 kinases if you include RET. All right? You can see at the top here on the left, plus means higher activity for the drug. You can see that AD1, 2, and 3 all hit RET well, but that doesn't correlate with their activity in the animal, as we discussed. Now let's look at AD1. It hits SRC, it hits BRAF, it hits mTOR. It hits all 3 pathways that our genetics say it has to hit, and that's why it works. Now let's look at AD2. AD2 hits SRC, it hits mTOR, but it leaves BRAF alone. Now, why would not hitting a target make a drug toxic? And this comes to a concept that Cagan and I call anti-targets, which are drug targets you actually want to get out of the drug because they're causing problems. They're doing what we call unbalancing the pharmacology. Okay? And what we, and actually a number of other labs, some of them before us, showed is that there's a feedback loop between mTOR and RAS signaling. Okay? So if you think about it, mTOR suppresses RAS pathways, for example, suppresses the effects of BRAF. If you fail to inhibit BRAF and you inhibit mTOR, you've removed the inhibition of BRAF and you have RET signaling through here. And now RAS signaling rises, not just in the tumor, and actually AD2 makes the tumor worse, but it rises throughout the animal. And we were able to show in a number of ways that AD2 is toxic because it's causing RAS signaling to rise. So if you were to think that all the way out, you'd realize, yes, Ross, that makes sense. If you inhibit mTOR, you don't inhibit BRAF, that pathway, the BRAF pathway, is going to turn up. Now we know that's causing the toxicity. Now how do we know this? Now we can turn to classical fly genetics. If we take AD2 and remove one copy of the downstream target of BRAF, ERK, so we just dial that pathway down a little bit, you can see now all the toxicity of AD2 goes away. Look at this. Now all the animals make it to pupation, and we even start to get some adults. Now AD2 starts to look a lot more like AD1. Okay? So if you think about it, that's a great concept, a great tool to use. We could actually walk through the fly kinome, protein by protein by protein, and just ask, in the presence of a knockdown of this protein, does the drug get better, worse, or stay the same? If we knock a target down in the presence of the drug and the drug works better, we would say that the drug needs to hit that harder. If we knock a target down and the drug becomes toxic, we would say we got to keep that activity out of the drug. Okay? And we can do that in a very controlled, rational way. So, for example, is AD2 as good as it gets? No. If we remove a copy from ERK from AD1, you can see, now that's a drug we would love to have. You can see that AD1 is not perfect. If we further balance the pharmacology, so we have it hit a better balance of RAF versus things like mTOR, that we can make a drug that really, if you're a fly, will cause all of you to live to adulthood. And these flies look so fine, we actually have to double check by outcrossing them to make sure they still have the cancer genes in them. On the other hand, I told you that hitting mTOR is bad. It's an anti-target. Suppose we did that in the presence of AD1. If we do that, what we find is that AD1 now starts to look like AD2. It becomes toxic. Okay? So if you didn't follow that, it doesn't matter. It's okay. The basic point here is we can stepwise map out what are the activities of any drug, what are the activities that need to be improved on by hitting harder, and what are the activities that are causing problems. And by using this simple procedure, we can rebalance drugs. And I have to say, we're doing this now for other drugs besides this, which I'll get to in a minute. So we went back to Cagan and Arvin. We said, please make a derivative of AD1, but it has to have the following properties. It has to continue to repress RET. It has to continue to repress SARC. It has to hit BRAF, but not too hard. And it has to leave mTOR alone. But you still have to hit that pathway. Remember, mTOR is in the PI3 kinase pathway. We know PI3 kinase is important. You have to hit that pathway. And so how do you do that? Well, we propose to hit the next kinase down, S6 kinase, okay? Which I'm showing here on the bottom left. mTOR actually activates S6 kinase, which is below the feedback loop. All right, so what do we have? They actually did this. They did some modeling. They showed if you add an extra charged groups out on the phenyl group, we could block the binding to mTOR, but still retain some of the other binding. We can get S6 activity and so on. And they sent us back two drugs, which I'll call AD1B and AD1C. And I hope you can see here on the right, AD1B and AD1C. Now these are the drugs we would really like to have. These are the drugs that, at least if you're a fly, will knock your tumor out without killing you. Remember, the left side, the black bars, are basically toxicity. The right bar is basically efficacy, and it's all good. Now let's look at its in vitro kinase profiles on the left here. Okay, let's look at AD1B. It hits RET, it hits SARC, but not too hard. It hits BRAF, but not too hard. It leaves the mTOR alone, which is good, and it hits S6 kinase to knock that pathway out. That's the magic. Okay? So are these drugs just great at treating flies? The answer, of course, is no. We have done an enormous amount of work in mammals. Let me just show you some of the data. If we take human cell lines and grow them in a dish, not a fan, but it tells us something. Vandetanib, we show that it actually works quite well to knock out an NZCRC1 human MEN2B cell line, which has an activating mutation in the extracellular domain of RET, in the intracellular domain of RET. And you can see vandetanib does a fine job in the micromolar range of knocking this out. However, 8080, which is actually AD1B, and 8057, which is AD181, I'm going to have to change those labels, they work about 500-fold better, okay, in a dish. Now, to be fair, bleach works even better, so that tells us something, but not everything. So we put those cells, or a similar cell line, the TT cell line, into a mouse to do xenograft studies. We didn't cheat. We actually grew the cells up in the mouse for 46 days to get a full-blown tumor of at least 100 millimeters. And then we asked, can the drugs reverse the tumor? This, of course, is more relevant to patients. You don't go into your doctor's office and say, I think I might be getting a tumor. Please give me something to prevent it. You actually go in the doctor's office and say, I have a tumor. Can you reverse it? So in that case, vandetanib fares very poorly in shrinking the tumor, but AD80 does a great job of knocking it down. Okay? And if you look at the individual tumors, many of them actually goes down, and sometimes I show the waterfall plot of that. And now the last question is, what about toxicity? Well, vandetanib has a well-known tox, which you can see here in the brown bar, the lower one. This is not labeled somehow. But the brown bar, the lower one, is vandetanib, and it has a toxicity if you just feed it to mice and weigh them. But AD80, or 81B, which is shown in the blue bars, is indistinguishable from controls, from DMSO, which is shown in the black bars. Basically, we can't find toxicity for these drugs at very high doses. And we think that's because we've really balanced the pharmacology. Okay? So the concept here, which I'm very excited about, which is shown here, is that we start with a fly that we've built a tumor model or any disease model in that we care about. We run it through our robotics to find an initial hit, and then we walk through the genome, either the kinome or if we have to, the entire genome. We identify targets and anti-targets that, when tweaked in the drug, would make the drug better in a whole animal setting. We come back to medicinal chemists and basically give them a list of how to change it. If they're good at their craft, and some like Cagan and Arvin are very good at their craft, they can use modeling design and then eventually build these better drugs. And then we come back to the whole animal systems and test them. And what my lab is doing now is we're actually using this approach to take known drugs, such as sorafenib, sunitinib, and so on. We've actually already mapped out what their problems are. We know these drugs have well-known toxicities. We've already mapped out what those problems are, and we're now working on creating what we call super sorafenib, super sunitinib, which are rebalanced chemical analogs that are, we hope, better in a whole animal and a whole person setting. Okay. And we're doing this for other diseases as well, but you know what? I need to move on here. Okay. You know what? I am going to show this. Okay. So I'm getting, time's running a little on, but I think I can go through this quickly, and then I do want to show you one other thing, which is complex models. Actually, you know what? I'm not going to show this. I'm going to skip over this, and if I have chance at the end, I'll talk about it. But I can summarize this. This is a concept I very much like, and it's summarized here. So what Derta Das in my lab is now doing is taking another, what I think is really a very unique approach. And he is screening drugs in flies, like always. But what he's beginning to focus on is not the tumor, but actually focusing on the normal tissue. So what do I mean by this? And I'll actually back up one here. You don't need to read this in detail, but just to give you an idea. He gives the fly the drug, the fly eats the drug, and then he pulls the normal tissue out as well as the tumor tissue. And he monitors actually 16 different readouts, which with the full colors you can see here. Okay? He monitors 16 different readouts, ERK signaling, RAS signaling, ECAD, and so on, Wnt signaling. We use mainly by running Westerns and using antibodies. And he basically, when he adds a first drug, he says, oh, okay, it suppresses certain targets, but then other ones in response come up. Okay? And in the normal tissue, you don't want things too far down, you don't want them too far up. That's called toxicity in these pathways. So the way he rebalances it is he looks at what goes up and down, and he gives a second drug to push the things up down. Okay? So now you have two drugs, one pushing here, but it's like whack-a-mole. You push these two down, another pathway pops up. You give a third drug, you push that down, and so on. And by the time you get to 3 or 4 drugs, if you're good at it, which Derta is, you can get to the point where you create a cocktail of drugs that are extremely non-toxic, far less toxic than any of the single drugs given at the same dose, but that are highly effective against the tumor, because tumors do not like having 3 and 4 anti-cancer drugs knocking them out. And let me show you an example here. Here is flies expressing a RET. And this, again, is rescuing them from lethality. Sorafenib rescues about 15% of flies to adulthood. Sorafenib plus bortezomib moves that curve up to about 25 to 30%. Sorafenib, bortezomib plus an HDAC inhibitor moves that up to 60%. And then if we add a PI3 kinase inhibitor, we can get to over 90% viability in that cocktail, again, at doses which are otherwise can be quite toxic. Okay? And this, I think, is a really exciting concept, which is, can we create complex drugs or drug cocktails where we are really focusing on the normal tissue, we keep things non-toxic so the patient can have a good quality of life, with the idea that the tumor, whose network is in some completely aberrant world, is not going to like that. It's going to get pushed into something it doesn't like. And you can knock the tumor out while maintaining the health of the patient. So this is something I'm very excited about. And we're now beginning to move into mammals to test these cocktails in mammals and ask the question, are they in fact less toxic in animals? We're mainly looking in mice and in pigs. Okay. What I want to really focus on instead, because my time is drawing short, is the flip side of what I've talked about. So I talked a lot about building complex drugs in a stepwise fashion using flies. But at the beginning, I waxed on about the importance of embracing complexity, the complexity that you see in the tumor. And so we are taking steps to do this. And what I'm showing you now is work by Erdem Bangi, a fabulous postdoc in my lab, who has really taken, I think, an interesting and unusual approach to going after colorectal cancers. So how do you model colorectal cancer? Now, this is something we can model in a fly because they do have a gut. And actually, molecularly speaking, as well as structurally, it looks a lot like ours. So what Erdem did is he went into the human patient sequencing databases, okay, and he asked, what is the most common double, triple, quadruple, quintuple, and so on combination of oncogenes and tumor suppressors that you see in individual tumors? Not adding them all up, but what's the most common combinations that you see together? For example, the most common quadruple combination that you'll see in a human patient, colorectal cancer patient, is RAS, P10, APC, P53. So he took that combination using transgenes and targeted that to the fly gut using magic fly tricks, and using more magic fly tricks, turned it on only in the adult. So he's creating tumors in the adult gut and just in one part of the gut, the so-called hindgut. Okay? In the case of RAS, he put in a transgene that activates it because it's an oncogene. In the case of P10, APC, and P53, he puts in a transgene that so-called knocks them down because they are tumor suppressors. Furthermore, not only, and I should say that the reason he didn't make a quintuple combination is simply because there are no currently sequenced patients that share 5 hits together because the statistics really fall off sharply. But as I'll explain, we can build 10-hit models as we need them. So not only did he build a 4-hit model, he built all the subset triples, all the subset doubles, all the singles, as I show here. So 15 different transgenic lines for every quadruple. And I should say he built 4 different quadruples for the 4 most common quadruples. Okay? I'm just showing you one example, the most common. So then he was able to ask the question, what do all these various things do in the context of other cancer genes? So for example, let me just show you. So he looked at 6 different phenotypes that he could quantitate. He looked at hyperproliferation, and what I'm showing you here is an image of the entire fly hindgut, so it's part of the gut, called the hindgut. And you can see here where the arrows are that there are polyp-like structures growing up out of here. That's because these cells are dedifferentiated or proliferating like mad. And so we see hyperproliferation, we see multi-layering. They're undergoing something called an epithelial to mesenchymal transition, and then they are migrating off to distant sites, which I'll show you in a moment. They also have a block in senescence in certain combinations, but not others. A block in apoptosis in certain combinations and not in others. And we could really see these emergent properties. Now, we all knew they were emergent properties, but it's really interesting to see them. And some of these emergent properties were very surprising. So here is another image, a close-up of a cross-section of a fly gut. At the bottom, that green tube is part of the fly gut. And I should say it's labeled with GFP, which is why it's green. To be clear, this 4-hit fly has 9 transgenes in it. Okay? That would be really tough to build into a mouse. So the green is the gut. The red is the muscle around the gut That blue that you can just barely see, that thin blue hose-like thing, is called a trachea. That carries oxygen. It's like our arterioles. And what you're seeing is a green cell that has actually walked out of the gut, pushed through the muscle wall, which you see is disrupted, has now corkscrewed itself around. So there's a single cell there, even though you see several parts of that cell along the trachea. It has corkscrewed itself around the trachea, so it's found its source of oxygen. It's now going to walk off to distant sites, into the abdomen, even into the head, into the wings, legs, and so on. And in some cases, it's going to form secondary tumors and kill the fly. Classic metastasis-like secondary tumors. When it hits those second sites, it's actually going to induce tracheogenesis into it, much like you see angiogenesis into our tumors, to build new sources of oxygen and build up those tumors. It's really remarkable how similar it is to what we see in people. Okay, so now here's the big question. What's the difference from a therapeutic standpoint between a one-hit fly, which is like the one-hit KRAS mouse that the field commonly uses, or a four-hit fly, in this case a RAS P53 P10 APC fly? So Erdem fed 16 drugs, mostly clinically relevant drugs. Most of them actually have been tried in colorectal cancer trials, all of which failed, all of which passed the mouse but failed in human trials. And he gave them to the one-hit fly. And as you can see here on the left, 13 of 16 of those drugs worked beautifully to knock the tumors out in flies. Okay? He then fed the 16 to the four-hit fly, and as you can see on the right, 0 of 16 of those drugs worked. All right? So this is why these drugs are failing in the clinics, because we're not accounting for the complexity. Now, Erdem had built all the subset triples, doubles, and singles, so he could actually map out what combinations were driving resistance in these flies. And so, for example, with BEZ235, which is a PI3 kinase inhibitor, and bortezomib, which is a proteasome inhibitor, by themselves, the quadruple was resistant to them. But what he was able to show is that pairwise, the two drugs were able to knock the tumor out. And in fact, they work by a very interesting mechanism in which bortezomib changes the network signaling of the tumor in a way that makes the tumor sensitive to BEZ235. And then BEZ235, which is a dual PI3 kinase inhibitor, then just knocks the tumor out. It's what we call induced oncogene addiction. And it's another concept we're very excited about. And this combination, we have an enormous amount of mammalian data. I think I have some here in a number of different models. I'll just show you one. This is not a fly model. This is actually a mouse colorectal cancer allograft, okay, of a triple mutation, RAS P10 APC. When we give it either drug alone, if you follow the colors, they are ineffective. But if we combine BEZ235 bortezomib together, and that's the green ones, you see all the green is shifted to the right. And interestingly, and exactly the way it was predicted in the flies, the lower dose of bortezomib actually works better. And that's true in flies, and it's true in mammals. And so the ones that work best is actually quite low. And if you're interested, I can talk about why that is. The mechanism by which these drugs work is novel and really interesting. But I need to move on. So the punchline of this section is complexity matters. And if you don't embrace and model that complexity, the drugs have the risk of combinations making the tumor resistant. Okay, and so this is basically my last slide, okay? And it is a slide discussing my center. Because to me, this is sort of the culmination of a lot of this work that we've been doing. And I should say, I think I said, but I'll say it again, that bortezomib BEZ235 combination is heading its way, or a similar combination is heading its way towards clinical trials. We've written a protocol and are waiting for its approval. And I probably didn't mention, but the AD80 and 81 were also picked up by a biotech company, and we're hoping that they will also get into clinical trials within the year or so. So flies are not perfect, but they have their use. And so we're now pushing this to sort of its natural next step, which is thinking about patient therapeutics. And so I began a center called Center for Personalized Cancer Therapeutics. And join with me, the co-directors are Marshall Posner, who is a clinician and scientist here, who's also who heads clinical trials for head and neck and is head of clinical trials at Mount Sinai. And Eric Schott, a PhD and professor here. He's also chair of the Department of Genetics and Genomics, and he heads the Institute for Genomics and Multiscale Biology. And Eric's specialty is, he is a brilliant, and Esquire magazine agrees on this point, he is a brilliant modeler and one of the top genomics people in the country. And so this has been really a powerhouse group at the top, myself excluded. And the idea here, which I'm showing you very simply here, is that pathologists come and remove the tumor. Well, the tumor is removed from the patient. Pathologists score the tumor, and I'll put names to this in a moment. The tumor comes out. If it is at least 50% purity, it gets sequenced, both for DNA, mainly whole exome, also for RNA-seq to get expression analysis and also to identify fusions and so on. The RNA-seq, that expression analysis, goes to Eric Schott's group, where a number of people do a network analysis and make both network predictions and even drug predictions. We actually take the whole exome data. So typically we see, and I should say we have 4 patients in the center right now. We just opened our doors a few weeks ago. We actually take all the mutations. So for example, one patient I was just going through this morning has about 140 mutations in their tumor. We actually take what are called the common variants that are the base ones like RAS and so on. We have flies already have that in. We take all the rest of the mutations, 135 or 138 mutations. We take the fly orthologs, we knock them down in the fly, and we ask, gene by gene by gene, functionally speaking, do they make that tumor better, worse, or no effect? That is, we're trying to identify drivers in a functional way, not a literature way, not a modeling way, just a functional way. And so for example, we just finished doing this passenger versus driver for a patient that has major thyroid carcinoma. We identified 8 different genes that, in addition to RET, are driving that tumor. And unfortunately, I didn't have the data when I submitted the slide deck, but I could show you pictures now. The difference between that rough eye and when you start putting those other mutations in, the eye is an absolute mess. The tumors are enormous, and it really, in many cases, just simply kills the fly from aggressive tumors. So we've been treating the RET fly. Now we're going to start treating this 9-hit fly. And so we do drug screens using our robotics. We do strictly FDA-approved drugs. We don't even bother with the single drug screen. We do 2 drug combinations. We just assume we're going to need a cocktail. And we look for drug combinations that work in the fly. If we get that, we then test them in mice. If we're able to successfully develop a PDX from the patient's tumor samples, we test it on that. In many cases, we can't do that, in which case we test it on a model that's as close as possible. Also, if need be, we test it in pigs for toxicity. They don't have tumors, but we can get a call on whole pig toxicity. They're closer to us. And based on that, we have 3 different committees that look through the scientific data, and also pharmacology committee that looks at the drug combinations we're recommending. They come back to the oncologist and make a recommendation for that patient. And what's nice is we actually have a series of drug combinations which we rank by various criteria. And so if the patient either doesn't work on the first one or it works for a while but then eventually becomes resistant to it, we have a set of other ones that we can also recommend as well. And here it's actually a large effort. This is just a small sampling of the people that are involved in this effort. A lot of people have really pitched in here at Mount Sinai to make this work. We don't know that this will be successful yet, but we expect that we're going to get better and better at this as time goes on. We'll also get faster and faster. And so I should say that we're taking medullary thyroid carcinoma patients, colorectal cancer patients, and triple-negative breast cancer patients for a number of reasons which I can talk about. We have chosen those 3. So with that, to summarize my talk, I told you about our efforts to build complex drugs through chemistry plus genetics to build what we call balanced polypharmacology drugs that hit many pathways but in a rational way. The second thing I told you about is our efforts to build complex models. And I should say with patients, we've developed new technology where we can very rapidly make a 12-hit fly, which we need for these patients because they often have up to 12 drivers. And finally, I finished by talking about the Center for Personalized Cancer Therapeutics and our effort to take these basic research tools and really try to get at the problems with the patient. And I should say, if we have one patient that's diabetic, we make the flies diabetic and then do the drug screen. If the patient's taking a particular drug, they often are, we give that at low doses to train the tumor, and then we do the drug screen. So we try to make these as close as possible to the patient. We don't claim that we understand the mechanisms going on. We just build as accurate a model as we can, and then we try to cure the fly. With that, I will stop. And this shows that to obtain continuing education credits, please go to this website. And I should also say that on at least the left side of my screen, if you have any questions for me, please just type it in the Q&A section, and I'll be happy to address it. Otherwise, thank you very much. Are there questions? No? Okay. How are we doing? Okay. I think I will end. Sorry, guys. I'm trying to figure out how this works. Oh, does it say question? Fantastic. Well, whoever sent the word fantastic, thank you very much. We're really trying to connect as much as we can with clinicians. For those of you who are clinicians, incidentally, we're very open to working with you. We're really trying to understand what are the issues with these patients. Meeting these patients has really taught us a lot about issues of toxicity and so on, and that's why we've begun to obsess over these, to try to build drugs that are more accurate. I should also say we're trying to combine this now with immunocancer therapy, coming in first with drugs to break the tumor up. We're hoping that it will make them more vulnerable to cancer approaches with Nina Barjois and working with us, and that too we'll see. So are there other questions? Okay, well, I will leave this on for another minute or two, and then I'll end. Anything else? All right. Listen, thank you very much, everybody, for tuning in, and good luck on your work.